The next Mendelian disorder that we are discussing is sickle cell anemia. As you have learned earlier, having two of the same alleles of a given gene means an individual is homozygous. If the alleles are not identical, then the individual is heterozygous. The sickle cell allele arose as a random mutation in the hemoglobin gene. Individuals who are homozygous for the sickle cell allele have sickle cell disease. Individuals who are homozygous for a normal hemoglobin allele do not have sickle cell disease but can contract malaria. Individuals who are heterozygous for the sickle cell allele are protected against malaria but do not get sickle cell disease. Heterozygous individuals have a mix of normal and abnormal hemoglobin molecules. Now let's see what is sickle cell anemia. Sickle cell anemia is an inherited blood disorder in which red blood cells or RBCs develop abnormally. Sickle cell anemia is caused by a mutation in a gene called hemoglobin beta or capital HBB located on chromosome 11. It is a recessive genetic disease, which means that both copies of the gene must contain a mutation for a person to have sickle cell anemia. If an individual has just one copy of the mutated gene, they are said to be a carrier of the sickle cell trait. If both parents are carriers, there is a chance for their child could be born with sickle cell anemia. The HBB gene codes for hemoglobin, a protein in red blood cells that carries oxygen around the body. A mutation in HBB results in a change in one of the bases in the DNA sequence from adenine to thymine. This then changes the amino acid, the hemoglobin protein, from glutamic acid to valine. This causes the body to produce a new form of hemoglobin called HbS, capital S, which behaves very differently to regular hemoglobin. HbS causes the red blood cells to develop abnormality and become sickle-shaped rather than the usual donut shape. They are harder and less flexible. This means that they can become stuck in the blood vessels, causing blockages. This is an account about the sickle cell anemia. I hope you understood. Now let's look at the next picture where you can see how the normal hemoglobin looks and how a clumped hemoglobin looks. Hemoglobin is made up of 4 chains. Clumped hemoglobin is causing the disease known as sickle cell anemia. Here is the Punnett square for sickle cell anemia. Look at the alleles. On top it is HB capital A and HB capital A. They are normal alleles. Normal alleles. HB capital A and HB capital A. Come to the side, it is HB capital A and HB capital S. HBAS is the genotype of the female. That means she is a carrier of sickle cell anemia. HB capital A is the genotype of the male who is a normal person. Now see the progenies. The first top two columns, 2 cells, are normal for hemoglobin. So no sickle cell anemia is found in that person. HB capital A stands for normal and no sickle cell anemia over there. Now you attend to the second row. The genotype of the progenies are HB capital S capital A. That means they are carriers. They are carriers for sickle cell anemia. Only one capital HB, capital S and capital S come together, then only that person becomes a patient of sickle cell anemia. In order to have such an occasion, both the parents must be heterozygous. Both the parents must have one allele HBA and the other allele HBS. Then only the progeny will become a sickle cell patient. You look at the next. In this Punnett square, you can see both the parents are heterozygous, that they are carriers of sickle cell trait. So what happens? In the first case, the progeny is homozygous for the allele and so not having sickle cell anemia. Second, and on the next row, third case, both are carriers of sickle cell allele, and the fourth one is really a deceased person with sickle cell anemia. Understand it clearly.